Microscopic evaluation of specimens. When specimens come down to the lab, we have to make the decision whether we should look at it under the microscope right away or whether it's something that we should first culture. So with certain types of specimens, and we'll go through different specimen types in here a little bit, it's going to be important to get some preliminary information up to the floor. Like if it's a cerebrospinal fluid that we detect bacteria in, then that person has meningitis, and that's some information that could save a patient's life. So getting the critical information or those critical results up to the floor is really important. Also, being able to determine whether a specimen has to be rejected or not. So if the specimen has too much contamination with healthy tissue or surrounding tissues or normal flora, we want to get that information up to the floor right away so that they know that they have to recollect the specimen immediately. Sometimes patients are discharged and we don't want to have them come back to the hospital to allow us to do those tests another time. So trying to catch all of those errors and catch all of the things that could deter a successful result and try to get the information up to the physicians as quickly as possible allows for better treatment outcomes. So when we get these specimens, if we're going to proceed with looking at it under the microscope right away, then there's direct examination. So the purpose of direct examination is it allows the laboratory to confirm the submitted material is representative. So sometimes if we get specimens and the specimen was taken from one of the healthier areas on the body, so if we have like a tissue that's necrotic and kind of nasty and it didn't include the necrotic portion, it just included the healthy portion, then that wouldn't be a representative material that was submitted. Identify cellular components and debris of inflammation to estimate the probability of infection. So if we smear and look at it under the microscope, and we can use some simple stains to be able to identify what's in the smear, but if we just take a cursory look at the smear and we see a lot of neutrophils, white blood cells that eat bacteria and eat substances and play a major role in mediating those the inflammatory response, then we could probably assume or safely assume that, yeah, there is an infection going on. Identify specific agents, so provide the physician with a presumptive microorganism identification. There's some areas of the body where if you take a swab from that area and you put it under the microscope and you see certain types of bacteria, you can diagnose them on the spot. Or you can give a presumptive diagnosis and tell the physician what they're dealing with. So for example, if a patient has a canker on their external genitalia and it's suspected that it's syphilis, if we get a swab sent down to the lab and we smear that swab on a slide, we can use phase contrast microscopy in order to visualize what's on the slide. And if we see spirochetes, then we can safely assume that the person has a syphilis infection. Treponema pallidum is the causative agent, so we would then call up to the floor or report the results as preliminary, but we can give them that information so that they know what antibiotics to start the treatment or to start the patient on for treatment. Guide physicians to early treatment with antibiotics. Again, that goes along with what I just said in the previous statement. So being able to give them that presumptive identification or that presumptive information can get the patient started on an antibiotic. Now, we don't know if it's the correct antibiotic and treatment might need to be changed down the road, but at least they're started on something. And develop epidemiologic data. So the preparation of samples. Bright field microscopy, specimen preparation must be in a manner that facilitates adequate examination. Specimens should be examined grossly to determine the best sample preparation approach. Thick but not opaque and thin monolayer smear areas should be produced by the smear process chosen. So in a bright field microscope, we've got light that comes from the bottom of the stage and it goes up through the specimen through into the oculars, and then it's magnified on the way. So it goes up from the bottom through the specimen into those objectives where it's magnified initially, and then through the oculars where it's magnified a second time, and then eventually into the eye. So because light has to go through the specimen that we look at has to be thin, and it has to be somewhat transparent so that we can look through it, or that at least light can get through it. If it's super thick, we're not going to be able to see through it, so we have to prepare specimens in a way that we can see through it. But that's not the only thing. We also have to consider, where is the infection going to be? So if we get a chunk of tissue, as I mentioned previously, there may be some parts of the tissue that look more healthy. There may be some parts that look really nasty and grody and black. And we want to be able to get representatives from the different areas within the sample. And the areas that are going to be more likely to be infected, we want to be able to look at those under the microscope. So preparing infected materials for visual examination. So for gross examination, a wet preparation can be performed. Typically, we look for parasites. Parasites and wet preps, we can also look for vaginal wet preps. So if a patient comes in with a suspected vaginal infection, we can take a swab, mix it with some saline, put it on a microscope slide, and we can identify within that specimen the presence of yeast, which Candida albicans is a pretty common cause of yeast infections. We can identify trichomoniasis, and trichomonas is a very common STD, but it's a small parasite that has a little flagella on it, and you can see them spinning around in the microscope if they're still alive. So as long as you get that specimen received onto the lab pretty quickly, we'll be able to see those spinning trichomonas organisms. And then we can also see clue cells. Clue cells are going to be regular epithelial cells, so skin cells, but they have a lot of bacteria in them, so their edges look kind of unclear and they don't have like a defined border. It kind of looks like their edges are blended in with the background because all of the bacteria are contaminating that cell. So you can kind of see all of that Gardnerella vaginalis bacteria on there, and that would indicate bacterial vaginosis. So if we see any of those clue cells, yeast cells, or the trich, we can get that information up to the floor right away, and they could start the patient on the correct treatment, because treating a bacterial infection, you'd use an antibiotic. Treating a fungal infection, like a yeast, you'd use an antifungal. Treating trichomoniasis, that's a parasite, so you'd use an antiprotozoan type medication. So very different types of medications, and we could get that information up to the floor quickly. So for microscopic examination, wet preparation, fluids or semi-solids, cytocentrifuged, clear or slightly turbid fluids, and then smears, you could do a drop, a pellet, rolled, or an imprint. An imprint, you basically just touch the tissue to the slide. So if you get a chunk of tissue, you just press it onto the slide. So smears from swabs. So if you have a swab from a patient, typically you want to collect 2 swabs. One of them is for direct smear, one is for culture. If we don't get 2 swabs sent down to the lab, typically we would try to mix the swab in some saline, and then we would use a dropper to put a drop on the microscope slide and then a drop on each one of the plates that we want to culture the bacteria onto. And that would, of course, dilute the bacteria, so that's not going to allow for a direct smear. That's kind of an indirect smear because we're diluting it in the saline, and the saline does have the potential to change what is inside of that specimen or how that specimen looks. Do not use swabs used to inoculate media to prepare a direct smear. Why? Because if you start smearing the swab on a plate, you are disrupting the integrity of the cells in there. And if there's something like epithelial cells, or if they're neutrophils or any of the white blood cells that are going to mediate the inflammation process, then you don't want to destroy them. You want to be able to see them under the microscope. So if you swab it on a plate and then you go smear it on a slide, you're going to disrupt the cells. Prepare smear by rolling the swab back and forth over the slide. Do not rub over the surface. It preserves the morphology and the relationship of the organisms, and it gets organisms off of both sides of the swab. So here you can see a sample of how you would just roll the swab on the slide. Smears from thick liquids or semi-solids. Best practices: immerse the swab in the specimen for several seconds. Prepare thin spread on a glass slide. Too thick is bad for staining, so you don't want to have a lot of organism. You're not going to be able to stain it, and then you're not going to be able to see through it, as we talked about with the bright field microscopy. Thin smears are adequate but may produce less desirable results than other methods. If you have super thin, you might not be able to see what you're looking for. So here shows how to smear from thick liquids or semi-solids. Smears from thick granular or mucoid materials. Best practices: smears should have both thick and thin areas. Granules in the material must be crushed to assess their makeup. Granules that are too hard to crush are probably not infectious material. The two-slide technique. So if you have something that's really thick or granular, you can put a sample of it on a slide, and then you can put another microscope slide on top of it to make kind of a sandwich, and you can rub those back and forth up against each other to try to break apart the substance that's in the center. And then you eventually remove that top slide, toss it in the biohazard, and make sure it's a biohazard broken glass container because it's glass. And then you can put a cover slip on it, you can stain it, you can do whatever you need to, but that would then grind apart and thin out that smear of what you're dealing with. So here's just a picture. You've got it on the specimen, or the specimen goes on the slide. Put another slide on top of it, smear it around, and then you can pull it off to spread it thin. Smears from thin fluids. So best practices: urine, cerebrospinal fluid, or other fluids you want to drop on the slide and mark the reverse of the slide. So you're going to put your little circle, your identifying circle, on the bottom of the slide, and you put the drop of the clear substance on the top. Do not spread unless it's too turbid. Why would these fluids be turbid? Well, if they have a lot of infection in them, so there's a lot of bacteria and white blood cells, or if there's a lot of protein that's found in the specimen or a lot of fat, it could create a cloudy environment. Also, like bloody specimens, if any of these are really bloody, that could create an issue. So do not spread it out unless it's too turbid, and turbid means cloudy. And then you're going to stain the slide, and cytocentrifuge preparations are the preferred method. So this just shows how you mark the bottom of the slide, you put the specimen on the top of the slide. Cytocentrifuge works well for preparing non-viscous fluids such as cerebrospinal fluid. Deposits the cellular elements and microorganisms as a monolayer onto the surface of a glass slide. Clears protein using a filter pad to clear the background. And it enhances morphology and concentrates the sample, making viewing faster. So a bowl is clipped to one side. The bowl contains a lid to close the sample. Then you place a small aliquot of the fluid, 0.1 to 0.2 mils, in the cytocentrifuge holder. You spin it for 10 minutes, and then you remove the slide and smear a portion if it's too thick. Fix sediment in 70% alcohol for 5 minutes. So here you can see when you cytocentrifuge, it's going to concentrate all of the cellular debris to a very specific area on the slide. And when it's done concentrating the debris in this cytocentrifuge method, then you can go ahead and smear it thinner if you need to. Staining for microscopic evaluation of cells. So the typical bacterial size is 0.4 to 2 micrometers. Like I mentioned, this class is going to focus on your typical bacteria. We're not going to go too much in detail on all of the parasites, the fungi, the anaerobes, or anything like that, because that's going to be reserved for our advanced microbiology class. So this class, focus on your typical bacteria. The typical microscopic shapes include cocci, or spherical, bacilli, or rod-shaped, and spirochetes, which are spiral. And then you can see different arrangements. So you can see cocci in clusters. Staphylococci would be cocci in clusters. Cocci in chains would be streptococci. So if you've ever heard of strep throat, streptococci, that's what it would look like under a microscope. You can also see them in pairs, so we would call that diplococci, or in tetrads. Just looking at the cell shape and staining the organism and referring to the source of where the organism came from, you can give a preliminary diagnosis or a presumptive identification to the doctor. Bacilli. Coccobacilli would be short rods. Bacillus of various sizes. There's fusiform bacilli. Palisading kind of forms these palisades here. And spirochetes, long spirals. Common stains used for microscopic visualization. So we use the gram stain the most often with bacteria. Acid-fast stain we would use if we're suspecting tuberculosis. So we'd do an acid-fast stain to potentially identify Mycobacterium species. Acridine orange would be another stain that we would use for certain procedures with the acid-fast stain. Calcofluor white we'd use for fungi. Methylene blue, this is our standard go-to stain if we just want to get a gross examination of cell shape. We can use methylene blue. Lactophenylcotton blue we would use with our mycology specimens. So lactophenylcotton blue is unique because it kills the mold spores. And we'll learn more about that staining procedure, and we'll actually get to perform it in advanced microbiology. India ink would be used to visualize the presence of a capsule. So if we have a very sick patient that is suspected to have meningitis and we get a cerebrospinal fluid specimen sent down to the lab, we would spin it down, smear it, and then we would use this negative staining technique because India ink is a negative stain. Negative stain means that it stains the background, it does not stain the organism. So the background would be stained, and then it would allow us to visualize whether or not the organism had a capsule. And if it did have a capsule from cerebrospinal fluid, we would be able to give a presumptive diagnosis or a presumptive ID of Cryptococcus neoformans, and we would call that up to the doctor. And then endospore stains. Endospore stains allow us to determine whether the organism is an endospore former or not. Stains can be classified as simple differential and probe mediated. Simple stains are just going to provide a stain and everything is going to look pretty much the same within that stain. Differential stains allow you to differentiate different structures or different cell types within a sample because they're going to stain different colors. So like, for example, an endospore stain, the endospores are going to stain usually green while the background cells are going to be pink. In a gram stain, gram positives are purple while gram negatives are pink or red. So differential stains allow for the different staining or staining organisms that are different, cells that are different, structures that are different, and you can see them visually by different colors. Probe-mediated stains are directed at specific organisms, and you can use antibodies that are labeled with particular stains or DNA probe stains. So the gram stain procedure, this should be a bit of a review of the procedure, but first step is to heat fix, but methanol can be used. So when we get the specimen on the slide, step one, we would want to kill the bacteria and cause it to stick to the slide. In the clinical lab, we don't use open flame for obvious reasons. We're in a hospital, we don't want fires, we don't want our oxygen lines that are helping our patients survive be exposed to flames. It's just a bad idea altogether to have open flames. Now we do have infrared incinerators that we could use, but more often we're just going to use a simple methanol because we don't want to have an infrared incinerator plugged in next to the sink, and the sink is where we're going to do all of our staining. So we douse it with methanol and let it sit. And we're going to let it sit for a full minute, and then we can rinse off that methanol and proceed with the staining procedure. We're going to add crystal violet, let it sit for a minute, rinse it. Then we're going to add iodine and let that sit for a minute and rinse it. New protocols that are coming out in the clinical labs say that you shouldn't rinse between steps of crystal violet and iodine. So sometimes it's just out of habit. I will not rinse in between those steps. So crystal violet and then douse the iodine directly on top of it. Then you're going to rinse with the alcohol or acetone. Alcohol or acetone serves as a decolorizer. And this step is very tricky because you have to get the correct amount of decolorizer done. You have to put it on for the correct amount of time, and you have to carefully watch it. And as soon as the purple stops running off the slide, you want to stop. If you over decolorize, everything's going to end up looking pink. If you under decolorize, everything's going to end up looking purple. So to get the correct gram stain, you have to time that and kind of get a feel for it. In this course, we're going to be doing gram stains as much as possible because the more gram staining experience you get now, the better you're going to be at it once you get out into your clinical placement or even into your career. Safranin. Safranin you're going to put on for 30 seconds. That's your counter stain. And then you're going to rinse that off and blot it dry with some bibulous paper and throw it on the microscope and look at it under the low power and then go all the way up to 100x with oil. This is a sample representative of gram-positive bacilli, and this is a sample representative of gram-negative bacilli. The gram stain may vary due to the crystal violet being rinsed too vigorously, so if you rinse off that crystal violet too much, after it's been applied, then it could wash away. The failure to use the iodine's a really important step because it makes that crystal violet adhere to the cells a little bit stronger. So if you don't use iodine, everything's going to be washed away and then everything's going to look pink in the end. Decolorizer is too vigorous or prolonged. So if you really douse it or let it sit on there, it's going to wash everything away and everything's going to eventually look pink. Insufficient decolorizing. So as I mentioned previously, if you don't decolorize enough, everything's going to look purple. Or if the safranin is applied for too long, that could impact the integrity of the cells. Usually, 30 seconds with the safranin and a quick rinse takes care of it. If you let it sit on there for too long, it could have some negative effects. So when you're studying this and you're preparing for not only this upcoming test, but also your qualifying exam, you want to know the ins and outs of the gram stain enough so that if I were to give you an error or something that you did wrong with the gram stain, you would be able to predict what the outcome would be. So I just gave you a bunch of examples of things that can go wrong and what the cells would actually look like in the end. You need to be able to read a scenario and figure out would the cells be clear, would they be pink, would they all be purple, would the cells not be there at all. So thinking through and learning this gram staining procedure enough so that you can answer those situational based questions is going to be important. And the morphology may be altered if the patient was put on antibiotics. So obviously antibiotics are going to kill bacteria. If they start on antibiotic treatment, the bacteria may look a little weird under the microscope. They may look like different shapes. They may look unhealthy. They may have broken open. So they may have a whole bunch of weird things going on with them. So that's just something to consider. You want to know whether the patient's been put on antibiotics or not. Acid fast staining procedure, we are going to walk through this procedure in advanced micro, but carbofuscin binds to the mycolic acid. And then there is the quinine stain. You cover with carbofuscin for 5 minutes, wash the slides with running tap water, decolorize with acid alcohol until the color is removed, wash in running tap water, flood the slides with methylene blue counterstain, and then wash with tap water to allow it to dry. The chalcofluor white stain. Chalcofluor white is a colorless dye that binds to cellulose and chitin. Chitin is found in fungal elements. So if you want to look at something under a microscope and you want to ignore the human tissue and illuminate the fungal elements, you would use the staining procedure. It fluoresces when it's exposed to long wavelength ultraviolet light. So you add a couple drops to the smear and then turn on the UV light. Or you can add it, let it sit, rinse it off, turn on the UV light and see if you see any fungal elements on there. The modified Wright-Giemza stain, so this is good for preparing blood specimens for the evaluation of parasites. So we may or may not perform this staining procedure when we get to advanced microbiology, but this is how we would identify malaria parasites in our Plasmodium. And there's different species of Plasmodium, and doing this staining procedure allows you to figure out which species is actually causing the issue. So we'll talk more about this stain when we get to advanced microbiology. And then this is just a good table that you can kind of look through if you're curious to learn a little bit more about the different staining procedures that we use. Microscopes. So once you stain it, then you got to put it on a microscope. The most common microscope that we use is the bright field microscope, but dark field can be used if we want to visualize spirochetes. There's also the phase contrast that will allow us to visualize those wet mounts. Dissecting microscopes are for large parasites, so if we want to identify a tick, the tick would come to the lab and then we would look at it under the dissecting microscope. Honestly, when I used to work at Aurora West Allis Medical Center, we would get bugs all the time sent down to the lab, even like spiders and all different types of insects, and everyone thought that they were ticks, and everyone was worried about getting Lyme disease. So when we receive the bug down to the lab, we try to identify what type of insect it is, and if it is a tick, then we have a manual where we just visually look at the picture of the ticks with its species and then look at the tick that was received, and then we can identify it. We can even identify the stage that the tick is in. So like early summer, they're going to be like the nymphs, the little tiny ticks, and those are kind of a pain in the butt because those are really small and difficult to detect. So if you do get one of those, you usually don't detect it until it is too late and it's been attached for an adequate amount of time for that Borrelia species to swap out and Lyme disease to be present. Later on in the summer, closer into the fall, you're going to see more of the adult ticks. And there are a lot of different tick species. Not every tick species carries the parasite that or the bacterium that causes Lyme disease. So being able to identify those using that dissecting microscope is important. Fluorescent microscope, there's certain applications. We can stain certain specimens with fluorescent antibodies or fluorescent DNA probes, and if it's positive for that identification, then our organism would glow. Electron microscope is very specialized, and it's usually used for non-culturable viruses. I don't really like to do a lot with viral identification in the lab other than molecular diagnostics with the viruses. So in your typical standard clinical lab, you're not usually going to use this type of microscope. Now, if you are working for like a health department or one of the reference laboratories, then you would be more likely to be working with identification of viruses. So here you can see how to observe different microbial pathogens and what the magnification is for the various areas. Terminology for direct examinations. Terminology is important for communication. We want to be able to communicate certain results across countries and across different computer systems, so we have to have a standardized set of language. We can describe microorganisms in a way that morphology can be used with specimen type and prevalence to imply the likelihood of a specific agent. So here's a table of the terminology. And this table, as we start to go through this entire course, is going to be really helpful because we're going to be dealing with a lot of the species that are on the list here. So if we know what shape to expect when we do the gram stain, we can go back and reference. Does it actually look like this? Is it gram-positive cocci tetrads? What is the more likely species that it would be if it looks like this? So keep this table handy. Go back and reference it as we go throughout identifying these different species throughout the course. And then here you can see a table of background material and what that background material might look like. Background material is, it's probably the most difficult thing to explain. You can only look at it and get to learn what is normal background, what's not normal background, and what is in the background. The first time I started looking at some actual smears of like pus samples, I was shocked at what it looked like. It just didn't look like anything that I would have ever imagined. So the more you look at specimens in practice, the more you're going to get used to identifying the different things in the background. So the examination of prepared material. In clinical infections, 10 to the 5th colony-forming units are commonly present in clinically evident infections. There's two types of infections. Monomicrobial are caused by a single species. Those are usually simple and straightforward, and they usually come from sterile sites. So if you get cerebrospinal fluid, there's probably only one organism in there causing the problem. Or if you get bacteria from a bladder infection, there's probably only one organism in there that's causing the infection. So monomicrobial would be from sites that are supposed to be sterile. Polymicrobial are caused by multiple species and they require more investigation. So if you have a patient that's been put on antibiotics and it kind of wiped out their normal flora, and now one species caused an infection, it increases the likelihood of a second species from being able to come in and cause an infection as well. So being able to identify the first pathogen and the second pathogen is kind of tough, and trying to figure out, you know, the fact that there is two pathogens or more than two pathogens is a difficult situation. It's more common in surgical wounds, aspiration pneumonias, perirectal abscesses, tubo-ovarian abscesses. Best practices to characterize the background materials: examine the material and note the characteristics, and then scan under low power. You want to determine if it's homogeneous or heterogeneous. Homogeneous means that it's the same throughout. Heterogeneous means that there's different portions with different types of looks to them. And then are the pathogens evenly distributed in only one field or in all of the fields? Is there evidence of contamination by normal biota? Is it necrotic? Is there evidence of purulence? Purulence is pus, so you'd see some white blood cells in there. Are there unexpected structures present? So searching for microorganisms. So when you look at it under low objective, first you want to determine what's the background, what's in the background. Is the background similar throughout, or are there differences? Then you want to determine, where are the microorganisms? Are the microorganisms in one spot? Are they all throughout? Are there a lot of them? Not so many of them? So when you start to look for the actual microorganisms, you want to scan for more detail at 40x or 60x objective. And you can use that 100x objective for a final evaluation. You definitely need to examine more than one area of the smear. You never want to put a smear on a microscope slide, look at one field and say, I don't see anything, and call it good. Because in an actual infection, sometimes bacteria are not present everywhere, or sometimes certain parts have more bacteria than other parts. So you may get to one of the parts that doesn't have any bacteria and think that there's nothing in the specimen, but scanning the entire slide allows you to then find it. You also want to consider what kind of site did this come from. If it came from a sterile site and you find any bacteria, it's significant because there shouldn't be any bacteria in a sterile site. If it came from a site that has normal flora or normal bacteria on the surface, then it's probably likely that there should be bacteria on there. Do not overinterpret the findings. There's strict criteria for microbial morphotypes that should be applied. So sometimes when you look at cellular debris in the background, you could be like, oh, this looks like cocci, and you could report out cocci when really there's nothing in that specimen that would be cocci. So when you're ever wondering what you're looking at, ask someone else. Ask for a second opinion. And you can wait for additional testing or staining if necessary. So sometimes, like for example, in a blood culture, if I got a blood culture and I think that there's gram-negative organisms in there, if I'm not 100% sure that I see gram-negative organisms in there, because gram-negative, they're red or pink. And what is the color of your blood? Red or pink. So they're really hard to detect amongst the red blood cells. If I'm not 100% sure, I'm going to want to verify that with, is there anything growing on the plate? Before I go and, tell the doctor or ordering physician, hey, there's something in here. I want to kind of double check. So not only confirming with another person, but maybe waiting for some additional tests to come back before we make the call. Grading or classifying materials. So the purpose, it prevents the culturing of normal flora or contaminated specimens. It prevents the use of antibiotics that may not be necessary. It's most important in sputum specimens. Sputum, remember that's that deep spit from the lungs. The criteria for separating samples before culture or evaluation. So there's different methods that can be used. And when you get into doing this in practice, these methods won't even be something that you have to memorize. It's just going to come second nature to you. So Bartlett's Q scoring, Murray-Washington method of assessment, and Hyman's method. So for the Q scoring one, you look for the quality of the clinical sample using the Q score. You want to determine the average number of neutrophils per low power field. And you can go up to here to see what the score is. Then you want to determine the average number of squamous epithelial cells per low power field. And you can go here to see what the score is. And then you do the calculation and do your interpretation. The higher the score, the better. So in a good sample, you want to make sure that you have a lot of neutrophils or a lot of white blood cells, but not a lot of epithelial cells. Why is that? So if I got a patient that comes in with a wound on their arm and their wound is broken open and I got to see what's causing the infection because this looks all infected and nasty. What I'm going to do is I'm going to take the swab, and although it's going to hurt the patient quite a bit, I'm going to try to get into the wound and scrape out the actual infectious material. Now, if I'm a nice nurse or a nice doctor, I may try to just scrape the surface just a little bit and send that specimen down to the lab. But being nice means that you're not going to actually be able to diagnose what's causing the infection. So, if the physician or nurse gets into the wound and actually scrapes out the infectious part, you're going to get pus, and pus has neutrophils in it. And neutrophils indicates that that is the area of infection. If the physician was quote unquote nice and they didn't want to hurt the patient, didn't want to get in there and get the actual infectious material, and they scrape it on the surface of the skin, well, what's on the surface of your skin? Epithelial cells. So you're going to see a lot of epithelial cells, which is not necessarily where the infection is. And when you look at those epithelial cells and you work up the culture, you're going to get normal flora. Probably a lot of Staphylococcus epidermidis. And when that comes back, that doesn't give you the information of what's actually causing the infection. So that's a major problem. So we want to make sure that there's a lot of neutrophils, not a lot of epithelial cells. Same thing with sputum. If you take a sputum specimen, you want to make sure that you're getting that deep infection from deep within the lungs. So when they hawk up that big spit that they're producing from their lungs, we want to make sure that there's a lot of white blood cells in there because that's where the infection is. And we want to make sure that there's not a lot of epithelial cells, because what lines the inside of your respiratory tract, what lines the inside of your mouth, that's epithelial cells. So if you just do a light spit into a cup, there's not going to be any neutrophils and you're going to have a ton of epithelial cells. If you get an actual good sputum specimen from the lungs and you cough that up, you're going to see a lot of pus. And that indicates that that's the area that has the infection. So the whole purpose of this is to prevent us from culturing something and working up a culture when in the end it's not going to be the actual infectious particle. So we want to make sure we get a good specimen of the infected area. We do not care about the normal flora. We don't care about what's on the surface of the skin or what's on the surface of your mouth. We care about what's actually in the lungs causing the infection, or what's actually in the wound causing the infection. So the criteria to determine the suitability of a sputum sample for culture, you can see we want group 4 or 5. So we want between 10 to 25 epithelial cells per low power field, or greater than 25 leukocytes. Or we can have less than 10 epithelial cells, greater than 25 leukocytes. So this is our ideal range if you're asked on an exam for this class, or if you're asked on the qualifying exam, what is a suitable range for epithelial cells and leukocytes, or white blood cells, neutrophils. For a sputum specimen, you would say less than 10 epithelial cells, greater than 10 leukocytes, or neutrophils. Sorry, less than 10 epithelial cells, greater than 25 leukocytes or neutrophils. So best practices, if you don't see that, reject the specimen and ask for a new one. And unfortunately, that's going to create some tension between the laboratory professionals and the nurses who have to face the patients and actually say, hey, we need to redo this specimen because it's not appropriate. Sometimes the people have left the hospital, they've been discharged, and now the nurse has to make a call and call them and tell them that they have to come back and redo it. And patients get really mad because it's a super huge inconvenience. It's not the laboratory personnel's fault. We're trying to get the best culture from the best specimen and make sure that we're doing the diagnostic procedures appropriately. It's not the physician or nurse's fault either. It's just sometimes these specimens are not appropriate for culture. So it creates for a little bit of tension between laboratory professionals and the nurses. You can also do the gram smear report. So quantitate the contaminating materials based on the amount of cells that you see. So 1+ would be light amount of cells, 4+ would be really heavy amount of cells. Those types of numbers are arbitrary, they're objective, or they're subjective, sorry. So you can take a look and one person may think, oh, that's a lot of cells, and another person may think it's not so many cells. So the more you work in the field, the better you're gonna get at making these calls. These types of notes are added into the patient's record directly. So if a specimen's sent down to microbiology, the tech literally types in, I see 1+ gram-positive cocci, or I see less than 10 epithelial cells. Like, those are things that get typed in. And some computer systems even have like drop-down boxes for where you would report those specific results. And then again, if you reject a specimen, you got to request a new culture. Important questions to consider. Do the materials fit within what should have been collected? So there's that grading here. Cellular fluid elements local to the sample. So in respiratory secretions, mucus, macrophages, goblet cells, ciliated columnar cells. In CSF, you should see some cellular elements. Cavity fluid, you should see some macrophages, mixed white blood cells, mesothelial cells. So if you see anything that's not normal or anything that's that shouldn't really be there, you can start to question, does this make sense? Did they collect it from the right specimen? Why are these missing? Or why isn't this here? Or what is this stuff in the background? So it's not just a matter of following procedure. And this is why microbiology really can never be 100% automated. It has to be evaluated by a person, because a person looks at the specimen and asks those types of questions. Does this make sense? And this main thing separates you from the computers and guarantees that you'll always have a job, is your ability to question things. And when you got into this field, you knew that this was a science field. You're going to be working in the laboratory, and scientists, that's the number one thing they do, is ask questions. So if something doesn't make sense or something doesn't look normal, ask why and do some investigating and do some digging. Don't just follow protocol and process results. Always think about what is going on, what's the specimen source, and does it make sense for the specimen source. Again, gram smear is quantified sometimes in certain situations. Culture identification guidelines, usual microbiota or brief presumptive description of colony type growth may be used. Antimicrobial susceptibility testing. No antimicrobial susceptibility testing is appropriate in certain situations. So if we have a urinary tract infection, we want to do antibiotic susceptibility testing. And in some situations, we just don't need to follow up with antibiotic susceptibility testing. Purulence criteria. So purulence, the presence of pus. We went through this a couple times already. Mucus and our heavy proteinaceous material may be present as well. The gram stain report, you would report it as the quantity, and contaminating materials should also be reported. Culture identification guidelines, so note the findings and correlate with the smears. Do they make sense? Do they match? Antibiotic susceptibility testing for Staph aureus, gram-negative bacilli, or others as appropriate. Mixed materials definition and criteria. The definition, it consists of these components all in a single smear. So if there's purulent, exudate, contaminating materials, and local materials, then you would follow the criteria below. Gram stain smear report, you quantitate only those organisms intimately associated with purulent exudates. So you only want to quantify the ones that are by the white blood cells. Must request a new specimen if the presence of purulence in uninterpretable culture results is found. Reports of direct examination, they should be made and sent out as soon as direct examination results are complete. Simple and include all elements needed for others to understand the report. So here you can see sample reports of direct microscopic examinations. So the source, sputum, expectorated. Microscopic, you can see heavy purulence, contaminating bacteria, yeast, and epithelial cells is heavy. And then you note gram-positive diplococci consistent with pneumococci. These sometimes are presented as drop-downs. So those are the options for you to be able to enter those into the computer. So you wouldn't necessarily write out all these words. You'd be selecting from drop-downs and sending up preliminary reports to the doctor. Initiation of special handling for unsuspected or special pathogens. So sometimes when we look at organisms under a microscope, we start to think, oh, this kind of looks like this type of organism, or maybe we should culture it on this just in case. So that way, we're not just following protocol, we're not just doing what we're supposed to be doing, we are also ensuring that we're not missing anything, that we're asking those hard questions to ask, and that we're thinking about things so that we don't miss anything. So sometimes if we think it's a slow grower, we may have to add, increase the incubation time. If we think it's a specialized type of microorganism, we may change up the media that we're using to culture it. Changing the temperature and atmosphere. So we may all of a sudden decide, well, this should probably be incubated in 5% carbon dioxide because I think that it's going to grow better at this temperature. If there's parasites seen, like let's say you're doing a routine culture on a stool specimen and all of a sudden a worm pops out of the stool, then you'd kind of pivot and start to do an examination for oven parasites. So again, not follow protocol, but also ask those questions. And if you see something that's not normal, you should definitely report it and pivot. Even as a simple phone call up to the physician and say, hey, I saw this, should we order this? And then kind of encouraging them to follow up with ordering add-on tests or additional tests.